Before going through some of the diverse ways that the hormone oxytocin can be released in the brain, it should be mentioned that there are other parts of the body that can synthesize oxytocin, typically for more local use, such as the thymus, pancreas, adrenal medulla, retina, corpus luteum, placenta, and testis. Most of the brain's oxytocin is produced in two hypothalamic nuclei. The paraventricular nuclei, a pair of these on either side of the third ventricle, and the supraoptic nucleus above the optic chiasma. The paraventricular nuclei release oxytocin, and to a lesser degree, the supraoptic nuclei. Both have magnocellular neurons, which project to the posterior pituitary, where oxytocin can be released into the bloodstream as a hormone. But the paraventricular nuclei also produce oxytocin in parvocellular neurons, which transport oxytocin along the axon and release it from vesicles at synapses with other neurons in the brainstem or spinal cord, and thus, in this case, oxytocin is a neurotransmitter. However, oxytocin can also be released from dendrites. This may be a widespread phenomenon that signals can be released from dendrites, but it is well studied using oxytocin from the dendrites in both the supraoptic and paraventricular nuclei. As released from dendrites, it acts locally as a paracrine signal, but can even affect the cells which secreted it as an autocrine signal. Finally, it seems that oxytocin can be released into the cerebrospinal fluid, and that cerebrospinal fluid can then transport it as a signal. This seems to be distinct from the signaling which involves plasma. This is because the levels of oxytocin in cerebrospinal fluid can change independently of levels in the plasma. The plasma oxytocin may have a half-life of a couple of minutes, while in the cerebrospinal fluid, its half-life can be a half an hour. The paraventricular nuclei border the third ventricle, and the supraoptic nucleus is near the subarachnoid space, and many neurons which perceive oxytocin with oxytocin receptors border the ventricles where cerebrospinal fluid is transported. And so therefore it seems that there are diverse ways in which oxytocin can be used as a signal even within the brain.